So thank you very much, Lara, and also Elena, for inviting me to be here. It's an honor for me to be at UNIMORE, and also it has been a pleasure during these weeks to be with all these wonderful students. And I'm trying to learn also a little bit from you during these days, and I hope you can also enjoy my seminars today. So if you allow me, I will start, and I want also to sorry the people who is listening from home, but I'm going to stand up. I'm sorry, but I cannot give a talk sit down. I'm sorry for that, but I think you will hear me, and you can see in the slides. Okay, let's go then. Perfect. So I think all of us know that we are living in a global world for the good things and for the bad things. The good thing for me is that today I'm here in Italy, enjoying good weather, nice people, talking about science during the lunch. And the bad thing is that due to this huge movement of people and movement of vegetables and plants, we have unfortunately many invasive pests. And invasive pests, as many of you may know, are causing huge damages to the global economy. Just one data. During the last 50 years, it has been considered that invasive pest has caused more than $1,003 billion to the economy. So imagine we are talking about huge damage that are causing these invasive species. And of course, we are talking about agriculture today. This is my field, agriculture, and where I feel comfortable. In agriculture, invasive pests are causing a lot of damage. And among the main pests are insects. And we scientists know that insects have been invading many countries during the last years. In fact, we can see in this graph how the number of invasive species is not stopping at all. It's increasing, as you can see here. During the last 100 years, there is an exponential increase of invasive arthropods pests. And unfortunately, it doesn't show any sign of saturation. So in other words, next 10 years, in the next 20 years, you may be here giving this talk, and you will still explain that there are many invasive species coming to Italy or Spain. And what is even worse, for Italians, Spaniards, French, people from Morocco, also in the north of Africa, we are in what the scientists call a hot spot for invasive species. What means hot spot? Means that because we are in the Mediterranean basin with fantastic weather, with great food, there is a huge movement of people and vegetables, and then we also have a good weather that allows the insect not only to arrive, but also to settle down here with respect to the Mediterranean countries. So that's why we are in a hot spot. And there is a crop that can explain very well that we are in a hot spot. This crop is citrus. This is a crop that I have been working during the last years. In fact, when I finished my PhD, or when I started my PhD in 2005, I started to record the number of invasive species that not only arrive to citrus, but also established and cause damages. So the one that I'm going to explain is not that they arrive to Spain. They arrive to Spain, they are causing damages. So during 20 years, we have almost 11 species of arthropods that are causing damages to people. In 2006, when I finished my PhD, this scale, Coccus magnolianum, started as a pest. It was after control by the biological population, but this, just to let you know, is one of the main pests in California. Fortunately, the state was controlled by a natural enemy. Just 2 years later, we discovered that Pesthiris sceliacus, a thrips, was causing damages to orange fruits. And this thrips was not controlled by any natural enemy. And it's still a big pest in Spain. 1 year later, just 1 year later, we discovered one of the worst pests that we have in Spain right now in citrus. It's called the Lotococcus abeliae. It's a millibug that has caused millions, or it's causing millions of euros per year. We do not have solution, or we didn't have solution until a few years ago. So during almost 20 years, it has been causing many damages to citrus. During that decade, we discovered also 2 species of the Tetranicus, 2 different species of mites. And quite interesting, we found them in the Valencia region, in the east of Spain, but also in Andalusia. The same insect in different areas. In 2014, we discovered one of the worst ones. Maistrello is the vector of HLV. This is likely the most dangerous pest that we may have in Spain. And in the last 5 years, or from 2015 until 2020, which I stopped there, we received 1 new pest every year. So imagine for someone like us working on invasive species, this is a tremendous work. And for the people, for the farmers, they are suffering a lot from these invasive species. And in my case, I work with invasive species, but I work mostly on biological control. And to work on biological control with invasive species is a great challenge. It's a great challenge, first of all, because some of these species we have no knowledge. Do you remember the millibug that I explained to you? It did not even know where it was coming from. It was totally unknown for science, this millibug. It was not considered a pest anywhere in the world. It was the first time that this millibug was discovered as a pest. We then discovered that it was coming from South Africa and started to study the biology and the ecology of this species. From the very beginning, to start a biological control program, you first need to know when it's causing the damages, when it's attacking the fruit, when it's moving to the roots, for example, in this case. So there is many knowledge that you need to know. And then you, of course, need to evaluate the native and the exotic natural enemies that you may use in a biological control program. That takes years. And the farmers need a solution, a fast solution. And this is something that we cannot provide with biological. These are 2 problems, but the biggest problem nowadays, and this is something that our governments needs to solve out, is the current international legislation. We have a big problem that are invasive species, but we have another big problem that the current international legislation does not allow to import natural enemies from other countries. That makes biological control extremely difficult for us. So that's something that our government needs to change. Well, today I'm going to explain 3 of the main pests that has caused damage in Spain. For the students, I'm sorry because I already explained to you one of them, but for the rest of the audience, I think it's important to know these examples. And I'm going to explain, first of all, about the biological control of the Lotococcus abeliae, the millibug that has caused so many damages in Spain. Then I will talk about Triotha erythrae, the civil vector of the disease HLV. And finally, a totally different thing when we talk about thrips. Okay? You can see that thrips, we have 4 species of thrips. This is something that I like to highlight. Thrips are super small insects that can hide anywhere. So it's very easy for them to move from one country to another country. That's why we have so many trips. Okay. So let's start with the mealybug, the Lotococcus aviariae. As I explained to you, we knew nothing about this mealybug. Nothing. And the first interesting thing that we found out when we were studying the biology of this mealybug is that, I don't know if you are aware, but most of the mealybugs can cause damages during the summer because they need high temperature. Mealybugs are best usually in tropical countries, in Mediterranean countries. They increase their population during the summer. Look what happened. This mealybug increased its populations during the spring. In May and June, the Lotococcus aviariae already increased its populations. And at that moment is when we have the flowers and the small fruits. And the mealybugs like to go to the fruit to feed on the phloem because there is a lot of nutrient phloem close to the fruits. So what was happening is that this mealybug fed on the phloem close to the fruits, as you can see in the picture here. When they feed the mealybugs, they also excrete some saliva, the same way that we digest, we use saliva. They do the same. But at that moment, this saliva is causing toxicity and deformates the plant. In fact, this is what is happening here. You can see in the second picture, this is a healthy orange, and you can see these oranges here. Will you buy these oranges in a supermarket? No way. Okay, totally weird oranges caused by this small mealybug feeding in the small fruits. Okay, and unfortunately, it's a very aggressive mealybug, and it causes up to 80% of the fruit damage. So that's why it's a totally different mealybug, because most of the mealybug cause damages during the summer, and most of the damage is because of the shooting. In this case, it's a much more aggressive mealybug. During our observations, we also find out that this invasive mealybug was attended by ants. That was quite curious, because this is a mealybug from South Africa, and it has just arrived to Spain. And what we found out is that studying up to 16 citrus oranges along the Valencian coast, we saw that this mealybug was attended always by the same ant species. This ant that you can see here is called Lasius grandis. So Lasius grandis was taking care of the mealybug. In fact, you can see here the mean number of mealybugs in these orchards, in these 16 orchards. We measure the abundance of the mealybug, but also the mean number of ants that were going up to the trees. Look, the highest the activity of the ant, the highest the abundance of the mealybug. This figure was explaining to us that there was a mutualism. Either we have more activity of ants because there are mealybugs, or we have more mealybugs because the ant activity is higher. We don't know. But there is showing us that there is a clear mutualism between a mealybug coming from the south of Africa with a Mediterranean ant species. So just after a few years, they have established this mutualism. So if the mealybug is attended by ants, what usually happens is that the parasitoids, the predators, cannot go into attack. They cannot attack the mealybug. Imagine you are 1 millimeter parasite. Will you try to attack that mealybug? If it's tended by ants, I can tell you, these ants are very aggressive. They will attack any insect that try to attack the mealybug, because they are feeding on the honey that is excreted by the mealybug. It's impossible for the natural enemies to attack this meal, or at least that's what we expected. And we ask ourselves whether we can improve the biological control of an invasive mealybug through the management of a native species. Okay, how can we do that? Avoiding the ants going into the colony, into the canopy, sorry, of the tree. What we did is to put glue in the base of the trees. So you can see here my colleague putting here some glue so the ants cannot go up, because the ants have the nest in the base of the trunk. So by putting glue here, the ants cannot go up. And we repeated this experiment in 2 commercial orchards. So these are commercial orchards. These do not belong to VIA or a university. These are commercial orchards where the farmer can do whatever they wanted. But we have different trees, you can see here in blue color, where the ants could go up. Control. They can do whatever they want. And we have trees where we excluded the ants. These are the ones that you can see in red color. Okay? Do what's okay. These are the number of ants as tending to the trees. Okay? We went there and we counted the number of ants during a couple of minutes. We went there and counting the number of ants that were moving up. So that means, for example, in February, no ants went up. In May, about 50 ants in 1 minute were moving up or down. Okay, this is relatively higher. So what you see with the orange color is normal. This is what happened in Spain, in oranges. The ants from May up to September are active and go up to the trees to collect the honey, to feed on the honey. That's a very common. We have done this experiment in other periods, and it's exactly the same. In other years, sorry, and it's exactly the same. When we excluded the ants, it worked quite well, except I think you will all of us remember very well what happened in March 2020. So we couldn't go there in a couple of months to exclude the ants, to put the glue, because it was COVID. We were not allowed to go out of home. So that's why during this period some ants could go up, because we could not put the glue on. Okay, but after that, it worked quite well. In other words, putting the glue, we could exclude. Okay, but the ants are not what we were interested in. We were interested in what was happening to the mealybug. Let's see when we exclude the ants. This is the number of mealybugs that we recorded in the tree. Not in the tree, of course. We cut some samples of the tree, some fruits from the tree, some leaves from the tree, bring back to the lab, and count the number of mealybugs. You can see in the control, you can see all the mealybugs went up in the spring, increased, and it was high during the summer. Look at the exclusion. The number of mealybugs in the canopy was much lower. So when we excluded the ants and reduced highly the number of mealybugs, if you remove the ants, you can improve a lot the biological control. Okay. This is for mealybugs and can be useful for other honey producer species. If you break the nutrients between ants and milkterans, you can improve the biology. In fact, for the farmer, what is more important is the percentage of fruit damage, right? Because at the end, we want to avoid fruit damage by the bees. And look what happened. We reduce almost half, or even more, the percentage of fruit damage by the mealybug when we remove that. So we improved the biological control, but it was more important for the farmer. We were improving or reducing the number of fruit damage. In case you are interested, we have published a couple of manuscripts describing first the interaction between the mealybug and the Mediterranean ant, and then how this is exclusion of ants improve the biological control of the mealybug. And sorry, I would like to highlight that this is part of the PhD of Ángel Plata, that will defend the PhD just in a couple of minutes. And then I move to the second test, and this is the most damaging. And as I said, I'm sorry to you because I already explained in the class the other week, but I think it's important for the rest of the Italian community that is following us today. And I'm going to talk about the biological control of Triota erythrae. Triota erythrae is known as the African psyllid vector. Sorry about that. It's known as the African psyllid. And it is well known because it's the vector of a serious disease called HLV. And HLV is a disease that kills fetus, and there is no solution against this disease. In fact, in Florida, HLV has devastated the citrus industry. It has nowadays. The citrus industry in Florida is lower than 80% that it used to be 15 years ago. And this psyllid, as you can see here, laid the eggs in the tender leaves. Then the nymphs develop in the young leaves and causes these damages to the leaves. This, compared to the disease, is not important. Important of this psyllid is that it transmits the disease. Okay? So this psyllid arrived to Madeira in 1992. From there, using molecular techniques, we know that it moved from Madeira to the Canary Islands in 2002. And from there, it went to the north of Spain, in the border between Spain and Portugal. And from there, it moved to the south of Portugal and through the north coast of Spain. So we know that just one entrance in Madeira moved to the Canary Islands and then to mainland. This is telling us that we should be very careful what is happening in the islands around Europe, okay, for the future. Anyhow, the psyllid arrived to Europe, and fortunately for us, the psyllid was without the disease. So in the same way that we have mosquito diseases, this psyllid arrived without the disease. Okay, that's the good news, right? But the disease can be anywhere in Europe, anywhere, because unfortunately, the disease is in Africa, Asia, and America. And unfortunately, many people move plants from one place to another place. Have you ever been in Asia, in Africa? And there are many beautiful plants. And unfortunately, the people take the plants back to Europe. This is something that should be totally forbidden. So that's why we think that even if we have not detected the disease in Europe, it may be. So it's very important to control the vector. Okay? So keeping that in mind, we try to carry out a classical biological control. The psyllid is originally from Southern Africa, and together with colleagues from South Africa, we searched there for parasitoids in different areas of South Africa. We moved first to the south of South Africa, and there we couldn't find nor the psyllid, neither the parasitoid. Then we moved to the north, and there we started to find different parasitoid species. In fact, we found Tamarixia dri that we expected, because the literature already said us that Tamarixia dri was the main parasitoid of the African citrus psyllid, so it was something known. But we also found Silaphus fulminatum, that is also a known parasitoid, but we also found a parasitoid that looked like Tamarixia, but it was not exactly the same. And in fact, using molecular techniques, we discovered a new species of parasite of the science. This parasitoid belongs to the genus Tamarixia, and unfortunately, we still don't know the species because it's a new species for science, and the taxonomies will need name for this new species. So we have two species of Tamarixia attacking Triotha. Which one should we import? Which one should we send to Spain to control the city? For that, what we did is we compared both parasitoid species in South Africa. We measured the parasites, we measured the abundance, but we also measured the sex rating. What psyllids are attacking and when they are laying eggs. And we found out that Tamarixia was attacking smaller psyllids than the newest species. You are able to attack smaller psyllids. That means you are able to attack different crystals. This is more interesting. So when we compare, it was clear that Tamarixia was attacking smaller psyllids than the new species. For that reason, we asked the permits to import Tamarixia into Spain. We asked the permits to the Spanish government, and the Spanish government allowed us to release the parasite, but only once, only in the Canary Islands. So we started our biological control program in the Canary Islands. We established a colony there with different isolines, something important. Do you know what are isolines? Imagine you have to bring a parasite, an insect from the other part of the world. You bring how many? 10? 20? 100? What we did is to establish isolines. Isolines mean I bring one parasitoid from one location. I bring another tiny parasitoid from another location. Then I came back to the same location, but 3 months later that it was much colder. I bring another parasitoid from each side. And these females are mating with their sons to establish isolines. So in this way, we are keeping the genetic diversity that we have in South Africa. Because if we bring the parasitoids put all together, at the end we will end up having a colony that is able to reproduce in the laboratory, but with a genetic background from the laboratory. That's something that we don't know. That's why we established different isolines. And then in April 2018, we released the first parasitoids. We did that in the Tenerife Islands. Do you know the Canary Islands? There are up to 7 different islands. Tenerife is the main one. The main island, sorry, is a volcanic island up to 4,000 meters high. Okay, small island, 40 kilometers, but very high. We released the parasitoid there, and it was quite interesting because just a couple of months after the first release, look at this leaf. This is my finger. You can see the size of the parasitoid here. It's quite interesting. This is not the lab, this is the product. So there is the field in Tenerife. Just a few months later, we already observed the parasitoid in the field. That was great news. That was great news. A few months later, do you remember the psyllid? The nymph? Look like healthy. They are totally dead. You can see the horse, right? That means that the parasitoid has emerged from there. So not the psyllid, but the parasitoid killed the psyllid and emerged from there. This picture, again, are from the field. So it seems the parasitoid was doing his job. And in fact, this is what you can see here in this map. This is Tenerife again, and with green color, you can see the presence of Tamarixia breed in the island of Tenerife. By itself, the parasitoid moved. You remember we released in just one orchard here. By itself, 1 millimeter moved up to 40 kilometers. And it didn't move to the south, because actually in the south, I explained to you, is a volcanic island. This is a super dry area where there is no Triotha. White color, no Triotha. In the south, didn't move because there is no triota. But in the north, they have a lot of triota. The parasite moved. In less than 1 year, they have moved more than 40 kilometers. But the surprise was not this one. The surprise came later. Because our colleagues from ICIA, from the Canary Islands, told us, look, Alejandro, we are finding the Maritzia drift in the surrounding islands. By itself, the parasite happened to the surrounding areas. 1 year later, it was not only distributed throughout Tenerife, but also to other islands. This is not the first case of a parasite that moved from one island to another island. It's happening in other parasitoids, and we really don't know how. We suspect that it moves with the wind, because in the Canary Islands there are different winds, and it changes. And also it changes depending on how high you are. Okay? So we think it's through the wind, but it can also be through the movement of plants among islands. Talking to the people to the Canary Islands, it's impossible. There's almost no movement of plants between islands. It's almost due to the wind. Okay, the parasite has established. Is it important? Yes. But what do we want? We want to control the seed. Is it controlling the seed? This is what you can see in the graph here. This is the proportion of citrus orchards in the Canary Islands. They also produce citrus that were infested with the seed. This was just 6 months after the first release. If you remember, we released the parasite in spring, and at fall, we measured how many of the orchards that we were sampling were infested. Almost 100%, between 90% and 70% in Tenerife, Gran Canaria, and La Palma were infested. Remember, we released the parasite only in Tenerife. 1 year later, look what happened in Tenerife. Less than 10% of the orchards were infested by the seed. In the other 2 islands, we still have the seed. Remember, we had not released the parasite. 2 years later, the seed had almost disappeared from the Canary Islands. It was less than 10% of the orchards that were in the Canary Islands, and the levels of the seeds in these orchards were extremely low. From there, the Spanish ministry allowed us to release the parasitoid in mainland Spain. And we established a new colony in Galicia, in Spain. Galicia is this area here in Spain. And from there, in Galicia, we released the parasitoid in Galicia, not from Spain, but also in Portugal. So the Portuguese colleagues also released the case. I'm going to explain what happened in mainland Spain in Galicia. In Galicia, they allowed us to release the parasite in 3 areas. But here, we do not produce citrus. You can see in this map here, we produce the citrus, like here in Italy, we produce the citrus mostly in Sicily, right? In Spain, we produce the citrus in Andalusia and in the Valencia region. But the seed is on the other area. It's doing there. What it's doing there is that in the backgrounds of the houses, most of the people have a lemon tree, a mandarin tree, just for farming consumption. All the houses in the Galicia has an orange tree or a lemon tree. And the seed has established throughout these citrus trees. Look here, the parasitoid, 6 months after the release. Remember, we released only in these 3 points. After, between 6 and 1 year later, it has spread more than 20 kilometers in mainland, without citrus orchard, just moving by itself from one orchard, from one garden to another garden, looking for a tree. It has a highly displaced movement. Then the government said, okay, you can release everywhere in Galicia, and you can see the red points are releasing points, and you can see the parasite established very well. But again, the important thing for us is to affect the city in mainland Spain, where it can be much more difficult. You can see it here. We divided the area in 3. Pontevedra, where we first released the parasitoid, A Coruña, and Lugo, that is this area here in front. So you can see here in Pontevedra, we have 28 points stamping point, and you can see it happened exactly the same that in the Canary Islands. 2 years after the release, we have control for the forest to have control. In A Coruña, exactly the same. We release 1 year later and control the snake 2 years later. And the same in blue. In fact, in 2023, sorry, we have been there in Galicia, and it's almost impossible to find the city. So it's a very low level. It's there. We know it. We know biological control does not control totally a pest. It doesn't happen in nature, but it has reduced a lot the density of the city. In fact, do you remember the leaves at the beginning of my talk? Do you remember this damage? These are all leaves. They are damaged, but the city is not there. Right? The same way that we have scars and they remain in our body, the same happens with the citrus tree. But look, the new leaves, they are totally clean. So you can see what happened a few years ago, and then that year was totally clean after the release. You can see here, the old leaves totally damaged, the new leaves are totally clean. Right? Same here, all leaves totally damaged by the city. But clean. In case you are interested, you know that you can read a little bit more about this project. But I cannot, I would say, I cannot finish with good news, unfortunately. I cannot finish with good news because this is the African city. And now I'm going to introduce you the Asian citrus tree. And unfortunately, 2 years ago, the Asian citrus tree that also transmits the disease has been found. First, it was discovered in Israel, and now this last year, sorry, in August, our colleagues from Cyprus discovered that the Asian citrus tree has also occurred. So that means that we have the pressure of the African citrus tree in the Iberian Peninsula, and now we have, sorry, the Asian citrus tree in the east part. So I have talked about the African, but the city is the Asian citrus tree is a much more aggressive tree. It works better under Mediterranean conditions. It transmits better the disease. So we are really concerned about Diaphorinaceae, the Asian citrus. And I think we Europeans, or Southern Europeans, Italian, Spanish, Greeks, colleagues from Cyprus should work in a project to control the city as soon as possible. So that's my message for the. And let's go with the last one. We change completely. So far we have seen to emit the lands, and now to move to the trees. And in the same way that you may have realized that I really like milligrams, or I really like interaction between milligrams and plants. I hate them. I hate them. Super difficult. Biological control tricks is a nightmare, or at least it's my nightmare, I would say. And as you can see here, we have now 4 tree species attacking our citrus. And this one, Canthanothrix orchidea, the Orchidea-Thrix, is a hard one. It's a hard one because, as you can see, it's damaging the fruit. It's just a cosmetic damage, actually. Just this damage. But in Spain and also in Italy, we produce citrus for fresh consumption. That means nobody is going to buy this. Nobody will buy this fruit here. It has the same flavor, it has the same smell, but just because of that, we cannot sell it. And this psyllid, this thrips, is unclear where it is originally from because you can see it's everywhere in the world. It's everywhere in the world. And in citrus, it's only in 2 places. Florida and Argentina. Do you know what happened in Florida? In Florida, it caused damage in 2 years. Then after 2 years, they had the Asian citrus tree. They didn't care about this. Because the thrips is damaging the fruit, but the seed was killing. So there is no any reference of what was happening in Florida with these thrips. From Argentina, we know that this is damaging a lot because it damages the fruit. Since the fruit is very small, up to harvest. This is actually what we saw in Spain. We saw that in June, more fruits, and the thrips start damaging the fruit and starts reproducing. And we have 3 generations throughout the summer. You can see this is the number of thrips per fruit throughout the summer. We have 1 pig, 2 pigs, and 3 pigs of nymphs and adults of the thrips. Both the immatures and the adults are in the fruits. The adults and the nymphs are in the fruit, but they pupate in the soil. According to our colleagues, these thrips pupate in the soil. That's quite interesting. Spend part of the life cycle in the canopy, but they pupate in the soil. And to measure that, that they pupate in the soil, we use this trap. Do you see this trap here? This is just a normal tube with here glue in the top and in the base. In the top, we capture the immatures that fall down to pupate, right? In the down part of the sticky trap, we capture the adults that try to fly away after pupation. Do you follow me? So we are capturing in the upper part the immatures that you see here, and in the down part of the trap, the adults. Look what happened with this. This is a common cut used for this. This is a very common, but the result was not so common. It was an unexpected result, I would say. And you can see clearly here what is happening. Look at the nymphs. We were capturing many nymphs throughout the year, but the adults were not emerging. They were dying. This technique has been used for thrips. It's a common technique. And usually you have similar numbers of adults and nymphs here. But in our case, the adults are not emerging. It's weird. Do you remember I told you it's spread everywhere in the world? The psyllid? It's spread everywhere in the world, but look. Here in America, in Central America, it seems there are more spots, right? So it seems this tree, this is what the specialists are telling us, is from tropical region. That means it needs a lot of humidity, right? If you are living in a tropical country, you are used to have very high humidity. So we had this. What if the thrips is emerging only in the areas that we are irrigating? Spain, like the south of Italy, water is super scarce. So we are using deep irrigation. Same here. I have seen some apple orchards here. You also use deep irrigation, right? So we thought that maybe the thrips during the summer is emerging only from these areas. So we use the same technique, but now putting them in normal soil and in humid areas. Look what happened. The emergence was much, much higher in the wet soil than in the dry soil. So this pest is actually only emerging from those areas that are humid, like in the tropical countries. So if we are talking about biological control, what can we do? We have to study the biology. Do you remember I explained to you? When we want to establish a biological control program, the first thing we need to do is biology. The biology has explained to us that this thrips is a pest because it emerged only from humid areas. So now, biological control, what do we do? who can survive very well in humid areas. Entomopathogenic fungi. Entomopathogenic fungi can survive very well in humid areas. Okay. So we wanted to test whether entomopathogenic fungi can be used to control these thrips in the humid areas. For that, came back to the lab and we tested whether the entomopathogenic fungi can attack these thrips. As you can see here, many of these that we tested, you can see here thrip mortality, and with different colors, you have different entomopathogenic fungi. And as you can see, two of them work very well and control perfectly the thrips under laboratory conditions. This happens with all the thrips and with most of the entomopathogenic fungi. Nothing new here. But we tested whether we could use it in the field. And we selected, of course, these two that work better. And we applied the fungi, as you can see here, only in the wet areas. Instead of spraying everywhere with an insecticide, we were just using entomopathogenic fungi in the low part of the trees. As you can see here, it worked quite well during June and July when we were using the entomopathogenic fungi. But during the August, when we stopped applying the entomopathogenic fungi, it didn't work. So this technique also is telling you that biological control doesn't work as well as we work or as we plan. We have studied biology, we have a plan, but so far it's still not perfect. We need to improve that to make it affordable for the farmer. We cannot tell the farmer, use entomopathogenic fungi every week, right? They will tell us, forget it, I'm not going to waste my money. We are trying now to improve this method to make economic. So that's how we are trying to improve the biology. With this, I finish this seminar. Just explain to you that every year we have an interest. If you want to join our lab, you are more than welcome, because I think we will have a lot of work in the coming years. And also, we have learned during these years that we have Emilia, we have an Emilia, and we have us. The best that we can do is to break these mutualisms to improve the biological control. And also, we have learned that with parasitoids, we can control some of these seeds. Not totally, but reduce a lot the population of seeds that are very dangerous for us. And I would like to finish just telling you that if we look for manuscripts, scientific manuscripts about biological control and citrus, look good. Since perfect, we are doing a lot of research. We are improving a lot of biological control of pests. This graph, we ask what countries are working on biological control? USA, China, and Brazil. Spain and Italy, we are not doing so well. Even though we are producing many cycles, we need to invest much money in biological controls. And with this, I finish, and I want to explain that all these biological control programs are not done by myself. It's done by a great group at IPVIA. As you can see here, we are many of us, and this is part of the PhD of several people that have been working really hard, and also from people abroad in South Africa, India, and the Canadians. On this, I finish this. Thank you very much.